Okay, good morning, everybody. I'm Stephen McLaughlin from the LMB Biophysics Facility, and today I'm going to be talking about analytical ultracentrifugation, which I'll refer to this entire talk as AUC. Okay, so let's think about biomolecules and what processes they can go through. Well, you could have, say, a molecule folding, a protein folding, and maybe it's binding, and it's binding to a partner, and perhaps you're going to crystallize something, or alternatively, the unfolded protein is going to aggregate. Alternatively, maybe proteins may kind of come together in complexes and join with other biomolecules to form like nucleosomes and form higher structures. So we can use AUC to look at the changes in shape and mass and solvent interactions. And to do this, then we need to spin the biomolecules really hard, so up to 60,000 RPM, to put a huge force on there so we can actually sediment them. We then have to, in some way, detect how they sediment and measure the time. We'll generate data, and to be able to then analyze that, we need to use hydrodynamic theory. But first of all, we'll go back into some history just to show you what AUC has kind of given to science so far. It's got a long history. It kind of started back here in the 1920s with V.S. Swedberg, and he was interested in colloid systems and gold, and he won the Nobel Prize in 1926. So his PhD student, Ferrer, said, well, why don't we try sedimenting some hemoglobin? It's red, maybe we can see it sedimenting. At the time, it was thought that hemoglobin was very small, given its iron content. But Ferrer said, oh, well, I'm going to put it in and try it anyway. And they kind of use these kind of AUCs, which are like the size of a room. Swedberg said, it's not going to work. I mean, I'm just going to go to bed. You can spin away. So in the middle of the night, Ferrer could actually start to see that the hemoglobin was beginning to be spun down from the meniscus of the liquid. He phoned up Swedberg and said, Thea, I can see a dawn. And that literally was the start of protein AUC. So way back in the 1920s, they did some formulation. Swedberg made the equation. His PhD student then went on to formulate the continuing equation. And they started to look at the molecular rate of proteins. And you must remember, back there in the 1920s, there wasn't any gels or anything like that. And people actually thought that proteins were some kind of colloids. And this kind of goes back to 19th century ideas. And it's quite funny to think now that we've almost come full circle to where kind of there's a huge interest now in protein composites. Swedberg then in the 1930s, could then show that actually proteins, the molecular weight, and they form subunits, and using hydrodynamic theory. And they looked at the size and the shape of the tobacco mosaic virus, and could then show that the very, very early EMs were actually depicting something that correlated with the shape and the size they saw by AUC. Again, in the 1940s, then we looked at shape of proteins. And then you probably, if you haven't come across AUC before, you'll know about the unit of AUC's S, Swedberg's. So, it was the discovery of ribosomes. So when we talk about the 30S and the 50S, that is all about sedimentation. And of course, the classic experiment by Mendel and Stahl on the semi-conservative replication of DNA, that was done with AUC. So in the 1960s, then there was a huge burst with Euphantis' seminal paper. And in the 70s, then with computer interactions, there could be, to analyze the AUC, you could then have the rebirth of hydrodynamics, and then started to look at really large polymerization. Now, in all this work here was done with these kind of large, machines, slightly smaller than a room, but they're still quite big. They were the Model E. And when it got to the 1980s, lots of these Model Es were basically falling apart, and they couldn't get replacements. So Beckman then made the XLI in the 1990s, and in the proteome. And then there was Tom Lau then made the fluorescence detection system. And then there's been lots of development in computer software for data analysis. So it's got a good path. What's the future? Well, Beckman have just introduced a new AUC in 2017 called the Optima, which has better data collection, more wavelengths, better camera. What do we use AUC for? Well, you can use it for anything. I mean, if you can sediment it, you can look at it. So there's a full range of things you can actually look at here. You know, why we want to use it? Well, a question might be, you know, well, what's your sample? Kind of get an idea of the mass of my sample. What's the shape and size? Is it interacting with the solvent components, i.e. detergents? We can look at associations, binding affinity, cooperativity, look at huge multi-complex systems, have an idea of, you know, what the kinetics of binding is, which we'll see just from the data analysis in a minute or two. What's the advantages? Well, it's in solution. There's no tags, usually, unless you're using a fluorescence system. It's a batch measurement. So the concentration you put in is the concentration that you measure. And you know, samples can be low. Hopefully we can cover the sample afterwards. It is unfortunately under vacuum. Sometimes the cells leak and sometimes the sample just gets lost. But that's a rare occasion if you kind of set your cells up right. And you know, as an illustration of, you know, currently what we use AUC for, here's a list of publications in the last couple of years from here at the LMB. And I'll actually talk about some of these examples later on in the talk to show you how we've used it. Okay, so first off, we need to get our sample into a rotor. Well, we're going to use special rotors, then over 4 or 8 hole rotors. And we're going to then put our sample in the sample sector here, which is then between 2 windows, either glass or sapphire. And then this all gets clamped into this cell assembly here. You torque it down, and there's 2 wee holes here where you can inject, say, a sample here and a reference here. And here's the 6th sector cell, which has got slightly different loading. Then you can centrifuge it up to about 60,000 RPM in these 4 rotors. And there's a variety of temperatures you can run things at. And you'll see what the use of temperature is later on. Okay, so how do we detect something that's going to be spinning? How do we detect when it's sedimenting? It's going around. Well, in absorbance detection system, we've got a xenon lamp, which is then shone directly through the sample. And then underneath, then there's an imaging system that then tracks across the cell as everything's spinning. And then the data comes out in a photomultiplier tube. So it looks directly through a reference and then the sample here. So if we flip this round here, because this is the center of the cell, so the center of the rotor, and then it's outside. So this is the going outwards. So if we flip this round and then we're looking outwards this way, this is the outer bit of the rotor, and this is the inner bit. So things are going to get centrifuged this way. So we'll have a sample sedimenting against a reference, and just simply by the Beer-Lambert law, then we can get an absorbance, and we can look at the concentration across the radius. There's another system we have, which is the interference detection system. It's using a laser diode source, and it has 2 slits here, and those fire the laser directly through the 2 sectors. There we go, through the sectors here. Then, you know, there's various mirrors and stuff below, and essentially it takes a whole image of on a camera across the entire cell. And so, of course, what the lenses do is they take the laser light that goes through the sectors, combine them in the camera, so that if you've got a slight phase change because there's a refractive index change within the sample, then when the light is brought back together again onto the imaging plate, you could get destructive interference. And if we look through other parts of the sample, then you may then get things in phase, so you get a bright stripe. So that when we look at this, something that's sedimenting, in buffer, you're seeing horizontal stripes. And then when there's a concentration gradient, it gives us an RI gradient, you then get bending of these stripes up. And the deflection then really depends upon the diffractive index over the wavelength. And for common proteins, that will then depend upon the increment of refractive index change against concentration.
which Chris was mentioning last week in his Mal's talk. And for normal kind of proteins, then we've got this kind of change against concentration in mg per mol. Now we're quite lucky at the LMB. We've got also the fluorescence detection system here, and essentially what this is a confocal alignment that sits above the cell, and we fire 488 laser through bounced off the dichroic mirror. Then it's focused onto a spot just slightly within the sample, and then the fluorescence comes back up through the dichroic mirror, is focused through a pinhole on the photomultiplier, and the whole array then scans across the cell as it's spinning. It's very, very sensitive, but of course you need a label or a GFT fusion, and it's also possible to look at samples in very complex medium such as serum, because you're only looking at the fluorescence of the label. Okay, so different detection regimes. What are you going to use? Well, the absorbance is quite sensitive. You've got a whole range of wavelengths. There is a scan time it's going to take you to scan across that cell. There is a limited range. Obviously, if you go up too high above the absorbance, then there's not very much transmitted light getting through. And if there's not enough material in there, then you might get very, very low absorbance. And so therefore, you're just going to be looking into the noise. But we can actually exploit the fact that with the xenon lamp, there's a peak at 230 in intensity. So you can actually exploit that to look at much lower concentrations of protein. Now with interference, then you can look at samples which you couldn't really look at in the absorbance system. It's got a very, very fast scan time. You can look at quite a good range of samples as long as you see these fringes being formed. But there is a limit where you will essentially reach hydrodynamic non-ideality, where you're basically at about 70 fringes per millimeter. So that's when the kind of normal equations we're going to talk about later kind of break down. So it's very good. It's very excellent position. Of course, because it's interference and it depends upon refractive index changes, there you've got to have care with buffers and baselines. If you remember back to the biosensor talk I gave about SPR. So it will see everything. So if you've got a buffer mismatch, then you're going to have some kind of background gradient. So it's very important to match your buffers. So with fluorescence, then it's very sensitive. Scan time is a bit quicker than the absorbance. You can have a large dynamic range, but of course you're limited by just the excitation at 488. So you need a fluorescent cell, but you can get away with quite small quantities. But normally when we're doing an AUC run, we would use absorbance and interference detection together, and then we would look at both of those data. And using those two systems in parallel has got advantages, as we'll see later on when we start to look at membrane proteins and glycoproteins. But that's for the last bit of the talk. So what kind of data are we going to get? We're going to spin something. So if we spin something really fast, then what's going to happen? So here's the meniscus, here's the bottom of the cell, here's going from inside the rotor to the outside of the rotor, and this is the concentration in the y-axis. So as we spin, things are then, the concentration is then going to be decreased here because you're sedimenting down. But if you do things really slowly, what happens then is you start to then just build up a kind of a stable profile of concentration against the radius here, and that doesn't change if you keep the same speed. And here's just a movie of what will actually happen. And you can see here's the meniscus here, and things are just then sedimenting all the way down to the bottom, and the concentration at the very, very bottom is increasing here. So that what we end up with, profiles like this over time. And you can see here that it starts off with the concentration is the same across the cell. Then at an intermediate time here, we seem to have got two kind of
kind of profiles here changing. And then near the end, then we've assumed we've only got one component kind of sedimenting. So if we look against time, against radius, then we've got this kind of profile here where there's something sedimenting quickly and something sedimenting much slowly. So how are we going to analyze this kind of data? Well, let's just walk through some of the physics behind it. You don't really have to worry about the maths or following all the details, but what's really important is some of the relationships between some of the parameters, which are kind of crucial to understand, you know, what the actual values you get out actually mean. So let's consider a particle in the middle here, sedimenting. So we've got a centrifugal force here going, you know, putting it to the bottom. That's going to depend upon the mass and then the angular velocity and you know, where it is in the radius. But that's going to be opposed by, well, by this buoyancy term here. And that's really the mass of the liquid of the solvent that this particle is then going to displace as it moves down. And that's separate then from this frictional component here. And that's kind of dragging, you know, the particle backwards. And that's dependent then just on the velocity and the frictional term here. So because we've got these different forces here, but we know that the particles are moving at a constant velocity, it must mean that the force of sedimentation must then be equal to then the buoyancy and the frictional force. So let's just, you know, put in what they actually mean. Let's rearrange them so we get the velocity on one side, and let's substitute in the mass of the solvent here. Now, the mass of the solvent is then will depend upon the mass of the protein, the density, and also this term called phi bar, which is the partial specific volume of the protein. And typically for proteins, then this is 0.72 to 0.74 mils per gram. And for DNA, it's slightly different. And if you remember back to Chris's talk last week, and if you haven't seen it already, please go back and look at it. He showed a plot of refractive index increments, that's the NDC, for all proteins on the x-axis, but on the y-axis was this term called the partial specific volume of protein. This can be calculated from amino acid sequences, and we'll see there's programs to do that later on. Okay, so let's substitute this in. And so we just then have got things in terms of the mass of the protein. Let's again rearrange. So let's get the frictional coefficient across here. And then let's get this omega r across. So this we're going to call this sedimentation, is then it's going to be equal to then the velocity of the sedimenting particle relative then to the angular velocity of the rotor. And this gives us the term Swedberg. And the Swedberg, the S value is equal to 1 × 10⁻13 seconds. And the thing to remember is that the sedimentation can be positive or negative. And of course, if you've got a negative sedimentation, what that means is something is floating to the top. Okay, so let's think that, okay, so this sedimentation is then directly proportional then to the mass of the particle, but it's inversely proportional to this frictional component. So what is this frictional component to do with? Well, the friction then depends upon, then this is Boltzmann's constants times the temperature, then in Kelvin, over the diffusion rate. And then that's dependent upon then the viscosity and the Stokes radius of the particle. I mean, for BSA, I mean, it's got a value of S value of 1.43 S. It sediments at this kind of rate at 50,000 RPM. So of course, the sedimentation, as you can think, is going to be dependent upon what velocity you sediment are at, and it's going to experience a frictional force of 0.05 femtonewtons. So, I mean, so this this frictional coefficient term we tend to then express relative to R naught. So it's a sphere of the same mass and density. And for globular proteins, this F over F naught is typically 1.2 to 1.5. So of course, if you measure a frictional coefficient outside these bounds, that's going to give you an idea of shape and size. Okay, so we've got these two terms here. Let's just put them together, where the kBt is really the gas constant over the Avogadro's number. Substitute in to this Swedberg equation here to give us then what's termed the Swedberg equation here. So you can see that the sedimentation coefficient is now also dependent upon temperature and the diffusion, as well as the mass. And this S value should be a constant. So if you have a particle then under ideal conditions, then it should be sedimenting the same in whichever country you do. And it's really relating then the diffusion against the mass. Right. Okay, so let's try and kind of think about, well, how are we going to actually solve this equation? How are we going to actually analyze our data? So we need to then think about fluxes. This is another way of thinking about it. If you think about flux, then really you're thinking about there's a flux of particles to the bottom that are sedimenting, but also there's that's opposed by a diffusion of proteins, molecules going back. So we have the flux in sedimentation is dependent on the concentration and the velocity, and of course the velocity is then is equal then to the sedimentation coefficient and the angular speed, and the diffusion then will oppose that, and that's dependent upon the rate of change of concentration differential against radius, and this diffusional constant, so that at any point in this radius, then the flux is going to be the differential between these two things here. So let's put them all together, and we get this is the flux, overall flux at a point R. So let's try and solve that. And essentially then we want to kind of get then substitute this JR term, and we can say that, well, okay, this dc over dr is essentially equal to 1 minus r over the flux at r over r. Substitute that in, and then we get this what's termed the Lamb equation here. That's relating then the change in concentration with radius, then against the diffusion, and also the speed and the sedimentation coefficient. But of course, if you look at it, you're kind of thinking, well, we can't really go any further in trying to derive that. There's not really a unique solution, but you can kind of reach a solution by just numerical and analytical methods of doing simulation against your data. So it's done essentially through number crunching then, really. So we have this profile here. So we can see what the sedimentation coefficient is going to be then by how fast this sedimentation profile, this boundary then moves down the cell. And of course, it's not a step function. You're not just getting, it's not a straight line, just kind of all the material here just sedimenting all the way to the bottom. There is a slope here, and that's due to the diffusion. And so then for number crunching in the computer, then we can end up then simulating a sedimentation coefficient, which would then give us this kind of profile. And we'll see how we do that in a minute or two. Okay, so let's think then about the other way of doing AUC, which is sedimentation equilibrium. So you're doing much slower speeds, and so this kind of sedimentation is then going to be opposed by diffusion, and so you're going to reach a time where really there's no flux happening. You've got a steady concentration profile here, because then the sedimentation and the diffusion are really opposing each other. They're equal, and there's no movement of this boundary. So we saw this then before, and then just the sedimentation flux is equal to the diffusional flux. So let's use the same equations again. We can put them in. And of course, this is just going to equal to 0, the flux at any one point. And then we can just do a little bit of maths to kind of say, well, you know, S of W over D is equal to 1 over C, where the DD ln C over DC is equal to 1 over C. Substitute that in. So we're going to get then, you know, SW over D is equal to this over D to the R squared. And if we solve that equation for a concentration at a point in the sedimentation profile, so that's concentration at position R, that's then going to come out as an exponential between, say, the point you're measuring and the initial point here. So let's substitute in this D value. Well, we know from Swedberg equation that, you know, S equals to dm over 1 minus these specific volume times the density over RT. Substitute that in, and now we've got an equation where we've got mass in there, so that now it's just a simple exponential now that is then going to depend upon the mass of the particle and also the speed that you actually move through. So you can imagine that what can you do? Well, if you change then the speed of centrifugation, then you're going to change this. You're going to change this profile, and then by fitting the exponentials to that, then you'll be able to work out what the mass is. And also, if you kind of look at this as well, you can see that we've taken out the diffusion, so there's no real shape dependence upon this profile here. It doesn't matter what the shape is. There's no frictional coefficient in there. But of course, we really have to then consider that proteins aren't really just dry spheres. Actually, there's always some water around there, solvent bound around there. So, the buoyant mass then is dependent upon the true mass of the protein, then 1 minus then the specific the V bar times the density. And so that's really about the mass that the mass of solvent that the protein then displaces. Okay, so but if we actually think about what's really happening, then there is bound material here. Then if we take account of that, then we've got these additional terms of this is the mass. And then this is the V bar of the protein, but then we've got these additional terms here, where this is then related to the bound water on the protein, and then the B3 then is about the co-solvents. But usually when we're doing sedimentation, we just can disregard these terms here because, well, the buffer is pretty much the same as water nearly. It's the density isn't really that much different, but we'll see later on where we can actually exploit the differences in, say, calculated mass against what the true mass is by taking account of these kind of terms in special cases. Okay, so how do we fit? I mean, the mass looks all a bit complicated, but actually, in practical terms, there's these lovely programs called SedFit and SedFat and UltraScan, where you can take your sedimentation data and put in the hydrodynamic parameters, and you'll fit your data, and you just hit go, and hopefully you'll actually get some good fitting. There's other programs here to analyze data. And there's a graphing program here called Gussie, which can also then analyze membrane proteins and glycoproteins, but we'll see that later on. So these kind of hydrodynamic parameters I kind of talk about, well, we can calculate that. This is a program called SedInterp, and you can then calculate from the amino acid sequence what the partial specific volume of the protein will be. And you can also then calculate the solvent density and also the viscosity. But we've got instruments upstairs here at the LMB where you can directly measure those parameters in terms of a viscometer and a densitometer. So if you've got an unusual buffer that's maybe not necessarily in the database, then you can measure those directly and put them into the program. If you've got a structure of your protein, then you can, or you can, if you want to make a model, or for AlphaFold, or look at two components coming together, you can actually use a hydrodynamic bead modeling from Jose Garcia de la Torre to actually calculate then in silico what the hydrodynamic parameters, such as frictional coefficient, could be. And sometimes this is a good way of just checking, are the fitted parameters that you get out from the AEC programs, is that compatible with what you get in silico? Okay, so let's think about doing some experiments. Well, what do you need to know first? Well, obviously, you need a nice pure sample, and you need to know what the amino acid sequence is to calculate the parameters. You need to know if there's any modifications. You need to have a defined buffer, because then you need to calculate the hydrodynamic parameters of viscosity and et cetera. So initially, what you might do is just do, with a sample, is just a hard spin down. It's called a velocity experiment, and do a CFS analysis to see, well, essentially, well, what's in your sample? Is it nice and pure, or is it it's just a whole mess of horrible stuff? Hopefully not. If say it's pure, then you can get some. You might want to look at interacting components, say. To look at, say, changes in distributions. You'd maybe label one component. If you want to get masses and fit binding constants, then you do sedimentation equilibrium. And obviously, good to do the velocity first and make sure that you know what components are interacting together. If, say, maybe you're not interested in interaction, you just want to look at your sample, you could say, well, okay, is the mass and the frictional coefficient reasonable? And then, is that tell me, it's one of the spurs, or do I really want to know what the mass is? And then go on to do sedimentation equilibrium. Okay, in velocity experiments, typically we would load then, say, different concentrations. And if you were looking at interacting systems, then you want to then vary one of the components, above and below the KD. You obviously want the static concentration component above the KD, because you want to then stoichiometrically titrate the interaction. The volumes in, say, these 12 millimeter 2-sector cells is about 400 microliters. If we're doing fluorescence detection, that's about 23 millimeter depth of sector, so you need 90 microliters. We can detect, with various ranges, and again using interference if you're doing absorbance. And for most complexes, then we'll just spin at 50,000, and collect 400 scans. If you start to get to big samples over 1 megadalton, then you need to kind of slow the speed down, and there's graphs to then calculate, what's the optimum speed. And then hopefully when you come to fit the data for CFS, for S distributions, you can get an idea of mass and shape, and then actually using sometimes in the KD between components. For equilibrium experiments, then if you're looking at you're doing just mass, and you load several concentrations, but if you wanted to look at, say, binding constants, then you would need to then, like with the velocity, have a range of concentrations above and below the KD. So you're actually populating the different species, so both the free and the complex ones. So we can do this in 2-sector cells where you use 160 microliters, or in these 6-sector ones where you can use a lot less. Again, similar detection, but we can't use fluorescence. So here we go. This is we do free speeds. We tend to do one for low, then we kind of go up for high, then to kind of deplete the meniscus, and we'll do a kind of a one at a very low speed to kind of baseline. And usually it takes, we do it with 12 scans with the hours between scans, so that can kind of take quite a long time, sometimes up to a week. And we can look at self-association, masses, equilibrium, and KDs. We'll see in some of the examples in a minute or two. So important considerations. Well, obviously, match the buffers. If you've got a reference buffer, especially for interference, make sure that the buffer itself, isn't actually absorbing. If you're going to use nucleotides, then that might then just completely absorb it in the UV region. So you're just going to have to look at interference data. You need a bit of salt in there, so you need at least 10 millimolar. Obviously, having concentrations, physiological concentrations where your sample is usually doing its action is probably best. Obviously, you're sedimenting, so keep the density of your solvent near as water as possible. I mean, if say you had a load of glycerol in there, what's going to happen? You're going to start to then create a density gradient, and that's really going to be very, very difficult to then analyze. So the buffer composition, again, if you've got things like DTT, swap that out for something like TSEP, because this will obviously oxidize over time. And then in the UV, you'll start to get a background kind of gradient. When you're using fluorescence, then there's a whole range of things you can, to get labeling. So you can use fluorescein on a fish tag, or you can use other labeling. Obviously, Alexidyes are much better. You can have a fusion. It's important to measure the degree of labeling, and remove any free dye, because obviously the free dye will then sediment as a kind of a very slopey background. And if you're looking at interactions, obviously always check that your interacting partner isn't just binding to the large hydrophobic dye. Okay, so here's an application. This is what we would kind of do. This is a project which a few years ago, which was looking at alpha-synuclein. There had been a paper which had said that if you purify alpha-synuclein from cells, it forms this tetramer. It was a clean sample, they said, and it was folded. While everybody else who has used alpha-synuclein, it's kind of the archetypal, intrinsically unfolded protein, though it may form structure when it comes into lipids. So the question then that the user had here was, well, is this alpha-synuclein that I've expressed in E. coli, can that form any kind of tetramer, at any concentration? So here's what the sedimentation profile looked like. This is a set fit output. It was in absorbance in 280. And so the different colored lines, you're going from black down to red is different times. You're putting a line here for where the meniscus is. The green are the bounds of the data you're going to fit. Here, this blue line here is where you think the bottom is. And then in this CSS distribution, then we have to then say, well, what is our limits of sedimentation? What do we think the frictional coefficient is? Let's put in some noise fitting. This is what we think the meniscus is. This is what our regularization is going to be. And then here's our hydrodynamic parameters. This is for the protein, which we calculated the V bar. And you put in the buffer density and the viscosity in here. And then we fit. And so as you can see, here's the solid lines of the fits to the data. And then we get an error bitmap here. And so this is going across each of the scans. So this is going downwards in scans. And if it's black or white, then that's where it doesn't fit, the fit doesn't fit to the data. And so if we see any structure in that, then we know that there's maybe something not kind of quite happening. If we collapse that all down, then we get a residual map here. And again, we're looking to see if there's any systematic variation. And so what we ended up with is we got a sedimentation coefficient here for the alpha-synuclein. And it was only one species. No matter what concentration we used, we only saw one species. There wasn't anything else up here. And we found it was sedimenting at 1.2. Now that was, if you think about what 1.2 is in terms of, it was a globular protein. Now that would seem to be quite small for what we think the alpha-synuclein mass is. But of course, what is happening is that it's unfolded and there's a huge, a much larger frictional coefficient, which is then giving us a much smaller sedimentation than we would expect given its mass. Now, the sedimentation here I've actually quoted in standard conditions, and that is calculated back if the sedimentation was done in water at 20 degrees. Because as we saw in the derivation of the equation, that this sedimentation can be then dependent upon both the temperature and also what the buffer composition is as well. So here's an example of an interference detection from the Laurie Passmore lab. And this was to kind of solve an issue where they solved this crystal structure, which looked like a tetramer, but other groups had said, no, it's a huge dodecamer. And you can see that, well, in the sedimentation, it looks a bit messy here. There's a lot of jitter here of the background, but this is time invariant baseline. So this really, if we can kind of solve that by doing some baseline, some error analysis, and we could then fit it. The majority then of the S value here was telling us it was a tetramer. There wasn't any indication of any kind of higher species. So, well, what happens, if you've got different kind of K-offs? Well, what kind of data will you can kind of get? Well, if you remember back to the NMR talks earlier on, then it's a similar kind of process that if you've got a very, very slow K-off, then when you sediment, say 2 interacting species, then you're just going to see the 2 components and then anything that's kind of binding. But if, say, you've got fast interconversion between, say, two binding species, then what you'll then get is an average sedimentation value that's then going between the 2 values here. We can exploit both these regimes, when we analyze data. So if you see kind of this with concentration, you can either distinct peaks, or if you've got kind of something that's gradually moving with concentration, you can kind of have some kind of indication of what the underlying kinetics is of the interactions. Now here's a first case of where we've got movement of the sedimentation profile here. So this is with David Rawn's group, and they were looking at this enzyme here, which is an amylase, which interacts on the ER chaperone BIP and amylase sit. And so this protein was found in 2 forms, either a monomer or a dimer. It was in a monomer. It then puts the MP group onto BIP, but in the dimer form, it actually takes it off. So this ratio of monomer to dimer is quite crucial for understanding its mechanism of then regulating of BIP. So different concentrations. Then as we increase the concentration, we can see a shift in the profile. And then if we integrate each of these, the peaks against concentration, we can see it increases here, we can fit a binding constant. And if we make a mutant, then it doesn't dimerize, then there's no change in the sedimentation coefficient. With concentration, sedimentation coefficient is pretty much at the monomer. Now here's an example of very slow kinetics, and this is from Paul Elliott, who's now in Oxford, and he's interested in this regulation of Lou-Bach-mediated signaling, which has involved ubiquitination and deubiquitination. And so this Sparta complex then binds onto HOIP, and then that binds onto this slide D and activates it. And so he wanted to know, was it actually really forming a complex? So sedimentation, so we could look at Sparta by itself, and this is slide D here by itself. And if you then start to add them together, you can see that the individual subunits disappear, and you start forming a complex with a higher S value. Okay, so typical applications then of sedimentation equilibrium would be, does my protein associate, or does it self-associate? Is there 2 components to associate? what a range of species are in my sample, what's the stoichiometry, is my binding weak or strong, I have a multi-component system, what binds to what. One question would be, is my complex fully formed? I mean, one of the, here's an example from the Loy group, where they're looking at the Arcadian dimer. And if you looked at it in sec-mols, because then you're getting dilution of that sample during gel filtration, then there's a whole kind of range of oligomeric states happening. At the lowest concentration, it was about 32 kilodaltons, and but the highest maybe it's at about 100. But it's interconverting, so it's difficult to assess. So I looked at this in AUC using this hybrid continuous analysis, where you essentially input the mass of the subunit and then ask, okay, if I have this mass of this one subunit, how can you fit all this data with concentration? So at high concentrations, then this data would fit with something that is actually an octamer. And if you decrease the concentration, then you see the other subunits populated. Using fluorescence detection systems, then you can look at hetero and homo organization. So if you label a component, you can see when things are very, very tight in the nanomolar range. And if you say want to look at hetero organization, you label one component, and then you can see that this label component then is into a higher form when you put more of the unlabeled components here, and you can fit Gaussians, integrate, calculate KD. So you can look at things that are what's binding to what. Here's a Magnetics study, which was looking at this this bicycle peptide, which was raised against the trimeric TNF alpha. So if we want to look at TNF alpha first, what we could see in the AUC was that it seemed to be, either forming monomers or trimers. But then when we put in then this bicycle peptide, you seem to get something that was intermediate. It looked like a dimer. So the question would be is, how is the bicycle doing it? Where is it shift? How is it shifting the equilibrium? Is it, say, binding to the trimer and disrupting that? Or is it binding to the dimer and stabilizing that? Or maybe it's just binding to the monomer and then just pulling this whole equilibrium across. So, okay, so if we label the TNF alpha, what we can actually see now is what's revealed is not only is there monomers and dimers, but there is actually a little bit of, sorry, monomers and trimers, there is a little bit here of dimers. And if we label then the bicycle, we could see exclusively that it's either unbound or just bound to the dimer form in the middle here. And then when they solved the crystal structure of the complex, they find that this bicycle then bound really near to the interface of the dimer, and this would actually then stop then the trimer actually forming. Okay, so let's in the last kind of 10 minutes just go to some advanced AUC. And so we're going to look at glycoproteins, and what we can do, if you remember back when the phi bar of the protein, we can calculate that. But what happens when you've got an oligosaccharide? Well, of course, that's going to have two things happening to the protein. Well, one, the phi bar is going to change because for oligosaccharides, the partial specific volume is slightly different. But if you're also adding all these glycoproteins onto the surface of a protein, well, what's that going to do? That's going to change the friction of the protein. So using this Gussie program, what we're going to do is we're going to have different concentrations of the protein. We're going to fit it, and then we're going to integrate sample here, put it into Gussie, and we put in the protein phi bar, then the buffer parameters of density and viscosity, the temperature, and we can put in what the extinction coefficient of the protein is. And so this is a method developed by Quan et al. And so what they found was, well, when you have different degrees of glycosylation, what happens is you've got the measured mass that you get from looking at the sedimentation coefficient, that decreases as you increase the amount of glycosylation, while you would expect here in black the theoretical mass to actually increase here. And so if we then fill in what the sedimentation coefficient is and what the frictional coefficient is, then we plot this, then what we then get out is a point here where the measured mass then interacts with what we think the theoretical mass. And that's going to give us a degree of carboxylation and give us the mass of the overall complex. And so this is then exploiting really a difference then of the partial specific volume of the protein against the partial specific volume of the carbohydrate, where this phi term then is related to the mass of the protein and the sedimentation of the glycoprotein, and also the diffusion of the protein here. So you can then calculate how much carboxylation you've actually got. Now for membrane proteins, it used to be very, very difficult because what people had to do was they had to match the buffer density against the either lipid or detergent density, so that really the density component, so the phi bar component of the bound detergent would just disappear in the calculations. So you have to, yeah, it's quite complicated. But Christina Bell developed this method where essentially you're just going to explicitly fit the detergent. So here we go, we've got membrane protein here, lots of detergent molecules bound, and you then get a peak here, which is due to detergent. And so explicitly then going to look at, you're going to need to then put in terms for the detergent or the micelle. So if we kind of think here, this is the equation here where this is the change in buoyant mass, and so this phi term, which would be then what the real phi bar is, partial specific protein of the whole protein detergent complex is. And so it's going to have a protein component, and then this other component here, which is the phi bar of the detergent, and this is the degree of then binding, or the fraction binding of the detergent to the protein. And so we need to be able to find out what this term is. So we just do so that we can then calculate what this real phi bar is. And to find out what this value is, we can exploit just the difference in signal between the absorbance and the interference, because proteins will absorb while the detergent doesn't, but in the interference, then everything, because it's a change in refractive index, it will give a signal for both the detergent and the protein. So here is, you input then the extinction coefficient of the protein, and this is the signal from the interference and the signal from the absorbance. And you need to know then what the change in refractive index against concentration is for the detergent and also the protein. Again, we need to kind of input those kind of values in. So we've got what for the protein is, got the density and the viscosity of the protein, and the extinction coefficient of the protein as well. What the mass of the protein is we put in. We need to know then what the phi bar of the detergent is, and we need to know the mass of the protein and the D and DC of both the components. And in GUSSI, then what we can do is there's various different ways we can actually fit it. One way we could do is we could find, calculate the mass against, say, different frictional coefficients, and then look at the plausibility. In the first case here, this is on the y-axis, we've got the calculated frictional coefficient here. And so in green is kind of what we think the middle, the calculated would be, and then the red would be kind of a higher limit, and the blue would be a lower limit. So, if we think, okay, well, if it's a monomer, then it's going to have a mass here, and this will give us a frictional coefficient of maybe about 1.3.
I say, okay, it's 1.29 within these kind of bounds of 1.3 and 1.35. Well, globular protein, you know, is that plausible? Well, that's pretty plausible. But what if it was a dimer? Then, well, if it was a dimer, then it would be slightly off scale here. Then the friction coefficient would be quite big, and you would think, well, that's not really. It's going to have a weird shape, but it's not really plausible for what we think a ball of detergent and protein is going to look up. Okay, another way of calculating would be from, you know, calculating the mass of the protein from just using S&D, or in this lower case, then we can look at the mass of the protein then using the sedimentation coefficient and then what we know about the Stokes radius. So say if we've done a gel filtration beforehand, and then we can estimate what the Stokes radius is, we can put that in, and then we can ask questions of, okay, we're going to calculate the theoretical mass for an oligomer, say, you know, if it's a monomer, a dimer, or a trimer, you know, well, we've kind of input then in sedimentation coefficient here, and we would then say, well, this is the data, what would correlate? And you can see here, this looks like a monomer. So we can look at the oligomeric state of membrane proteins. So, okay, that was velocity. So what about sedimentation equilibrium? Well, let's go back to our Cadent dimer. If we spin this at 3 different speeds, at different concentrations, and fit it, then we can fit to, say, a single exponential. And then this gives us an average mass. And we find that it was this mass here, which was very, very close to the octomeric species. And here's an example of using sedimentation equilibrium, which I did very recently for Elise Fisher in the Barfoot group, which was to solve a problem about stoichiometry. She's looking at these checkpoint components. Then this is MAD, and this is a small part of BUB. And the crystal structure, she found a very symmetrical complex where you know, 2 BUBs then bind to this dimer of MAD. But when she looked in the ITC, then when she was titrating in the sample, then that's titrating in BUB into a dimer of MAD, she found that the stoichiometry was actually 1. So that means that 1 BUB was then binding to the MAD dimer. Well, that didn't seem to work out. So, you know, is this just a crystal artifact then? Is this symmetrical? So we've used AUC and sedimentation equilibrium at different concentrations. So we're looking at MAD alone, and then in complex. And then when we looked at the mass, we could find that the mass of the MAD alone was pretty much as expected. And then we added various concentrations of BUB. We really got masses that were really close to the expected for then a 2-to-1 interaction, i.e. that's going to be an asymmetrical complex where only 1 BUB binds to the site. So therefore, I mean, what was happening then, the reason why this crystallized very nicely was because it was symmetrical, but in solution, this coil is asymmetrical, and it only has 1 binding site then for BUB. Okay, and also you can say, look at self-association. You can look at, you know, calculate the free monomer and the monomer and the dimer. And then you can calculate then what your binding constant should be. And here's an example from Mark van Bruegel, where this was a component where, is it actually, again, a crystal artifact, or is it a real dimer? He also made a mutant at the interface. And even though it was a very, very weak dimer, we could actually see that it was binding. And when you make that mutant, then you get a shift here, which means that really it's not forming a dimer anymore, and it's monomeric. Of course, then you can do various models then to look at heterosociation, and in SEDFAT there's a huge variety to be able to fit. And you can exploit differences in absorbance between DNA and to do multi-signal fitting. So in a question here would be, well, say if we label the component, or we look at the absorbance, say of a fluorophore, then we could just then look at what the fluorophore is doing. And then if we looked at interference, and that would just then measure the protein, because if this is quite very small, then you may not see it in interference.
And then you can calculate then the KD. So in summary then, AUC, it's really easy to do. All you have to spin, you can computer analysis, that might be a bit complicated. Because you can spin anything, then it's available to a wide range of biomolecules. We can answer really simple questions in composition, self-association complexes, binding constants, give us an idea of masses and shape. We could go into really complex kind of mechanisms of action and tackle, difficult questions with membrane proteins and glycoproteins. Further information, there's various books, and on AUC particularly, and in this recent book here, there's a chapter on looking at AUC. There's various websites. Obviously, Beckman is one of the suppliers, or other suppliers of AUCs. You can go to look at the software by Peter Schuck and SedFit, or Ultrascan by Doris Delimmer. And there's a website here to all questions about AUC. And for people in the LMB, then there's more information on the internal website. And I'll just use you a few of, we got a dedicated room in the LMB with AUCs here. And if you need any advice, just drop us an email. So thank you and goodbye.